Scientists have altered the genetics of one bacteria to create way more of a new antibiotic called formicamycin. You might be wondering, what's so special about it? Well, in order to answer that, first we need to get into a bit of history. Antibiotics have been around for a very long time, probably since the origin of bacteria 3.5 billion years ago. And this is because bacteria naturally produce them. We tend to think of antibiotics starting with Alexander Fleming's discovery of penicillin in 1928 and its subsequent use in the medical field around 10 years later, making penicillin the first natural antibiotic. However, even 20 years before this, in 1909, scientists were messing around with some toxic arsenic compounds and created the first synthetic antibiotic called salvarsan. Because penicillin was first discovered in mold, people typically think of mold as being the source of antibiotics, when actually, most antibiotics used clinically are from bacteria. Especially from the genus Streptomyces, which makes more than half of the antibiotics we use today. This genus truly is the MVP of antibiotics. We might also think this because these bacteria are literally named with the suffix myces, which means fungi in Greek. So these scientists must have been a bit confused when classifying these organisms. They saw that these bacteria made filaments and were like, yo, that's a fungus. But it actually wasn't. It didn't have a nucleus or other fungal defining features, so it was a bacteria? Oops. So wait, you're telling me that bacteria make the very thing that kills them? Yes, but one type of bacteria will make a compound to kill another type of bacteria. But it's more than that. Antibiotics can be used as both weapons as well as signaling molecules to communicate with other organisms around them. In smaller doses, it can be used to say hey to another bacteria, but in larger doses, it can be used to destroy all who oppose them. You probably know that antibiotic resistance is a major problem, and if you didn't know, antibiotic resistance is a major problem. So we need new antibiotics, but there has been less incentive to be in the antibiotic discovery field lately. This is for multiple reasons. One being that the scientists doing these high throughput screens kept finding the same compounds, and they were like, well, I guess we found them all already. But now we are realizing that bacteria probably don't act the same way in the lab as they do in their natural environment. I mean, this is relatable, right? I don't act the same way in a job interview versus when I'm at home.
It was skin. The tuberculosis is caused by bacteria called Microbiology.
So why would we expect these bacteria to act the same way in this plastic block? When I say they don't act the same way, I mean that they probably aren't expressing the same genes, synthesizing the same compounds, or talking to their neighbors as if they were in the soil just chilling. So antibiotics are naturally produced by bacteria from clusters of genes called biosynthetic gene clusters. These gene clusters encode all the necessary components to create these compounds. It's like having a meal prep kit. Everything you need is all in one package. From genomic analysis, we only think about 10% of these biosynthetic gene clusters are activated in a laboratory setting. So there are many more antibiotics for us to discover. A team of scientists recently teased apart the regulation of one such gene cluster from the species Streptomyces formicae, which creates the compound formicamycin, which is a rare, pentangular, polyketide antibiotic. Oof, that's a lot. Scientists actually found this compound 10 years ago, but despite its efficacy against several strains of antibiotic-resistant bacteria, it wasn't a viable antibiotic because they could not make the bacteria produce enough of it. A couple years later, these scientists were messing around with the 24 genes in this biosynthetic gene cluster by specifically upregulating the activator and downregulating the inhibitor, which caused the bacteria to create a lot of formicamycin. Wow, yay science! Additional cool side note, this bacterial species was found in a harmonious relationship with the ant that lives on a leaf that cultivates its own fungi garden, and the bacteria just hangs out with them. So it's a leaf, ant, fungi, bacteria mutualism community. Cool. Besides that little co-op situation that they have going on there, what can we learn from this? For starters, we might want to rethink the way we treat our bacteria because that soil really must hit different. Thanks for watching. Have a bioZone day.